Good morning, everyone. My name is Kelly, and I'm a first-year PhD student in the biomedical engineering program. And my background actually isn't in biomedical engineering. I started off as a biology pre-med student and then thought, well, I like math. Maybe I want to take some math classes to fill in free electives. And I got into doing biomath. So I'm going to tell you a little bit about mathematical modeling, which is like the heart of biomath. Is anyone familiar with mathematical modeling? Kind of, sort of. So I know everyone here is a science major, so do any of you actually like math? A little bit here and there. Well, I'm going to tell you a little story about the research that I did and how mathematics and biology can go hand in hand and really help each other out. So I did a mathematics postbac program where I was doing only math for the past year, and I was able to get onto a biomath project, which is something new that was starting up at Smith College. So there was a scientist, neuroscientist, his name is Michael Barisi, and he worked a lot with zebrafish. So he has the biggest zebrafish research lab in the world. And one thing he was working on in particular was the KIF11 mutation. So KIF11 is a motor protein that assists in pulling chromatids apart during mitosis. So what happens when KIF11 is deficient, when there's a mutation, is that everyone's familiar with. And while that's not like a biologically sound picture, you get stuck in mitosis, and that's what it is. So what Michael did, his students in his research lab were physically counting on slides the number of cells that were stuck in mitosis in a wild-type cell that didn't have the mutation and in a mutant cell. So Michael collects, well, his poor students count hundreds of cells and get all of this data. What Michael comes up with, so this is time on the x-axis and cells in mitosis on the y-axis. So over time, in the wild-type cell, cells enter into mitosis, and then eventually they leave. So then it gets to be a sort of decline by the time that you have a fully developed zebrafish, which only takes about 3 days or 72 hours. With the mutant-type cell, you get something that looks like this. So this is the number of cells in mitosis, and we know that the mutation is leading to an arrest in mitosis. What do you think is responsible for this shape of graph? Any ideas? The cells are entering into mitosis, then they're just accumulating. So one thing Michael thought was, well, if the cells are just accumulating, why is there a decline, and why is there this? It should look a little more round, a little bit more smooth. So this is where he got stuck in his project. And he brought it to my research advisor, Nessie Tania, who worked strictly in the mathematics department and just started picking up a little bit of mathematical biology. So he said to her, I don't know if accumulation is the only thing that happens, and I really want to understand this deficiency, so could you help me out a little bit? Do you think you could model this? So this is where Nessie picked it up, and this is where we're going to get that intro into mathematical modeling. So the first thing that you want to do when you are starting a model is to come up with a schematic, which is just a very basic way of laying out the biology so that you can come up with a differential equation to kind of understand what's going on. So that's the bio side. Now we'll get into the math side. So one thing that's going on here, we have two populations of cells. We have glial cells, which are cells that are going to enter into mitosis, and we have cells that are in mitosis. So the first thing we're going to work with is the wild-type cell, and that's the one that can enter into mitosis and exit out of mitosis. So the first thing we have is a population, or a rate, and we'll call that Rn, of cells that are becoming glial cells. Follow that? So then glial cells enter into mitosis, and they're going to enter at a certain rate. We'll call it Rm. Now, the cells in mitosis, because this is a normally functioning cell, they can become progenitor cells. We'll call that RP at at some rate RP. Or these cells in mitosis can become glial cells. So we'll call that RG. That's the very basic schematic. So now we're going to go into the mutant cell. So again, we have a population of glial cells, we have a population of cells in mitosis. Does anyone have any idea of where we might start with this? What rate we're going to put in? So it's very similar to the wild type. So we still have a population of cells coming in, and we still have glial cells that enter into mitosis because the deficiency doesn't have an effect until cells are in mitosis. So we'll call that RM again. But now, are we going to have the same rates that we had before with cells leaving mitosis and becoming progenitors? No. And what do you think what's going to happen to those cells? They die. So those cells in mitosis, because they're just arrested in mitosis, they're going to die, which is going to cause destruction of the fish at about, again, 72 hours post-fertilization. Fish is cooked, it's done. So we'll call that death rate KD. So now we're going to come up with differential equation to explain what's going on in our schematic. And we'll just, for the sake of time, we're just going to work with the mutant model. So we have two populations of cells, and those are the two cells that we want to model in this case. So we have glial cells, and we have cells in mitosis. So DG dt, this is going to be our glial cells at a certain point in time. So now we're focusing on this schematic right here, and we're going to come up with an equation for that. So our glial cells. We have a positive number of cells that are becoming glial cells, and that's equivalent to Rn, the rate that cells are becoming glial cells. So that's positive. But now, glial cells are leaving when they enter into mitosis, so that's a negative effect on the number of glial cells. And that's proportional to the rate that cells are becoming glial cells. So that's going to be negative, the rate that cells are going into mitosis, times the number of glial cells. Does this right here make sense, this interaction? Okay. So now we have the cells in mitosis. So we have dm dt. Anyone want to give this a shot? Or at least one of the terms in this equation? Don't be shy. So we have glial cells that are becoming cells in mitosis, and they're going to mitosis at a rate of Rm. So we have G times Rm. But now there's a negative effect again. And that negative term is the number of cells in mitosis that are dying at a rate Kd. So we have M times Kd. So this is where you start, and this is a very basic differential equation set that Nessie used to help out Michael. So she looked at these equations, and she played around with MATLAB, and you sort of do a little bit of curve fitting. So one of the things she thought, Michael's initial hypothesis was that cells are just accumulating. The problem when cells are just accumulating is that your data looks like this, which doesn't fit this curve here. There's just too many cells in mitosis. So she thought, okay, maybe we increase the death rate. Maybe cells are just dying after they're accumulating for a little while. And the problem with that is that your curve looks like this. The cells die off too quickly. So she did a little bit more fitting, and what she found was a curve that looked like that, which fits the data a lot more nicely. And she told Michael that she got this when she decreased the rate at which cells were entering into mitosis. But when we talked about KIP11, that didn't have an effect until cells were already in mitosis. So Michael said to her, I don't believe you, because the cells are entering in they should be entering in at the same rate. This shouldn't have an effect on this. But she convinced him to set up an experiment that took like 3 months to perform, and he tracked the time that cells were entering into mitosis and found that there was a really, really big delay in the cells going into mitosis, and that's where I picked up on my research, and that's what I had worked on for the test. That's all. Any questions?
Any comments on the presentation? think it was nice. You told the story and taught us a lot of things within the story, and it's good.
You did a really good job of breaking it down so that I
Did you
could
guys follow
understand.
that?
Yes, and I'm not really a math person, so.
That's what everyone, so when I gave this talk, I gave it at a math presentation, and I had to like take out all of the biology, like this was the only thing that we threw out there because otherwise it was too much bio.
I liked your ability to move on whenever you asked questions and no one was really responding, rather than getting hung up on that. One suggestion would just be to allow a little bit more time. Like, I know it feels like a lot of time has passed, but I don't think it ever really went more than about a second for a possible response.
You seem very confident.
You're a good math teacher.
Thank you.
All right, thank you.